Welcome back to AllAboutRx. We're jumping right into glycogen metabolism today, an absolute must-know topic for your pharmacy and med biochemistry exams. We're going to cover everything from how we build glycogen to how we break it down. And of course, those high-yield clinical pearls you literally need to know to ace your tests. If you've ever felt totally overwhelmed by all these enzymes and pathways, don't sweat it. We're breaking the whole shebang down step by step in this explainer.
All right, here's our roadmap. We'll kick things off with a quick introduction, move through glycogenesis and glycogenolysis, look at tissue fate, dive into regulation and pathology, and then wrap it all up with a rapid-fire exam summary.
So, let's start with the basics of glycogen.
Glycogen is, essentially, this massive, highly branched polymer, and it's built entirely from alpha-D-glucose molecules. Now, this whole process happens specifically in the cytosol of your liver and skeletal muscle cells. But, it doesn't happen for free. It takes an initial energy investment. We need ATP to phosphorylate the glucose, and UTP to activate it so it's primed and ready to be added to a growing chain.
Moving on to building that store, glycogenesis.
When mapping out this entire transformation, starting with glucose 6-phosphate and ultimately ending up with a highly branched glycogen structure, it's really just a beautiful, logical progression. It's a step-by-step journey from preparation right through to activation, elongation, and finally, branching.
Let's zoom in on those first couple of steps. Things kick off when the enzyme phosphoglucomutase converts glucose 6-phosphate into glucose 1-phosphate. It's basically just moving that phosphate group over. And that brings us to a highly testable moment, the committed step. The enzyme UDP-glucose pyrophosphorylase, or just UGPase for short, uses UTP to form UDP-glucose. Boom! Once that UDP-glucose is formed, the molecule is fully activated and totally locked into the glycogenesis pathway.
But here's a pretty wild biochemical quirk. The main builder enzyme, glycogen synthase, it actually can't start a brand new chain from scratch. It needs an existing chain to add on to. So if your cellular glycogen stores are totally wiped out, how do we even begin? Well, a protein called glycogenin steps up as the essential primer. It literally builds a short starter chain by attaching glucose directly to its own tyrosine hydroxyl group. We call that autoglucosylation.
Once that primer is laid down, glycogen synthase takes over as the master builder. It elongates the chains linearly, taking glucose from our activated UDP-glucose and linking it up with alpha-1-4 bonds. But honestly, a single long, straight chain just isn't very efficient. Enter the branching enzyme, or 4,6-transglucosides. This guy snips off a 6 to 8 residue segment from the end and sticks it onto the interior of the chain, creating a crucial alpha-1-6 branch point. Why do we even care about branching? Because doing this over and over creates tons of non-reducing ends. That means when your body needs energy right now, multiple enzymes can chew up the glycogen from different ends all at once for super rapid mobilization.
Next up, glycogenolysis, breaking it all down.
Now, this degradation pathway introduces a massive high-yield exam pearl. Glycogenolysis uses a completely separate set of enzymes. Seriously, it is absolutely not just the reverse of synthesis. We're dealing with totally different molecular machines to dismantle the branches we just worked so hard to build.
The whole dismantling process starts with glycogen phosphorylase. It aggressively chops those linear alpha-1-4 bonds, popping off glucose 1-phosphate molecules one by one. But it has a huge limitation. It hits a structural roadblock exactly 4 residues before a branch point. It just physically can't get any closer. When it stalls out like that, it leaves behind this stubby branch structure that we call a limit dextrin.
So to clear that roadblock and deal with the limit dextrin, the bifunctional debranching enzyme swoops in like a specialized cleanup crew. It actually has 2 distinct activities. First, its 4,4-transglucosidase activity grabs the outer 3 residues of that stubby branch and shifts them to the end of a different linear chain. That exposes the final single-branched residue. Then, its second activity, amylo alpha-1-6 glucosidase, comes in and snips that final alpha-1-6 bond. That specific snip hydrolyzes the residue and releases a beautiful molecule of completely free glucose.
Okay, let's talk tissue fates, liver versus muscle.
The really crucial thing here is understanding where this newly freed energy actually goes, and that entirely depends on the tissue. This is incredibly high yield for exams. The liver has an enzyme called glucose 6-phosphatase. This lets the liver convert glucose 6-phosphate into free glucose, which it generously pumps right into the bloodstream to feed the whole body.
Muscle cells, on the other hand, they completely lack glucose 6-phosphatase. They're totally selfish. Any glycogen broken down in the muscle stays in the muscle. The glucose 6-phosphate just goes straight into glycolysis to power your workout, and zero free glucose makes it to the blood.
Moving on to regulation and pathology.
This whole system is a perfect balancing act, mostly run by two hormones. After a big meal, when your blood sugar is high, insulin steps up. It drives the building of glycogen stores, increasing glycogenesis, and slams the brakes on glycogenolysis. On the flip side, when you're fasting, glucagon takes the wheel. It flips the switch, signaling for rapid glycogen breakdown and halting synthesis completely.
So let's apply this for a second. What do you think happens clinically if the liver's primary enzyme for releasing free glucose, that glucose 6-phosphatase we just talked about, is genetically missing? Think about what the liver is supposed to be doing for you during a fast.
A deficiency in glucose 6-phosphatase causes Glycogen Storage Disease Type 1A. You probably know it as von Gierke disease. Without this enzyme, the liver is completely unable to maintain your blood sugar levels during a fast. The glycogen gets broken down into glucose 6-phosphate, sure, but it gets permanently trapped there. Patients present with severe, life-threatening fasting hypoglycemia, all because that one vital step to release free glucose into the blood is broken.
Let's bring it home with our rapid-fire exam summary.
Take a mental picture of this rapid recall checklist because it perfectly contrasts the specific enzymes and key molecules for building and breaking glycogen. Glycogen synthasase pairs with alpha-1,4 synthasase, while glycogen phosphorylase handles the alpha-1,4 breakdown. Do not forget the primer glycogenin, the activated UDP-glucose donor, and of course, the limit dextrin stall product. Lock these in and you'll instantly spot the right answers on test A.
To leave you with a bit of a provocative thought, if you really grasp these pathways, how do you think a branching enzyme defect would look under a microscope compared to a debranching enzyme defect? Think about the structure of the glycogen that would pile up in each situation. One would be long, fragile, and unbranched, while the other would be a massive, clustered mess of short, stubby limit dextrins. Ponder that as you review your notes. Thanks for studying with AllAboutRx. Make sure to like, subscribe, and check the description for links to complete study notes. See you in our next explainer.